Inside every one of your cells, a hidden microscopic world is constantly at work. What you are seeing is DNA replication. Billions of molecular machines racing along your genetic code, perfectly copying the instructions for life. And this is real-time speed. This is exactly how fast it happens, inside you, right now. But copying the code is only part of the story. How does the cell decide which instructions to actually use? This is the start of a gene for a bone-strength protein, waiting, silent, locked inside the strand. A molecule of vitamin D arrives. It binds to a receptor, and together they attach to the DNA. This is the trigger, gene activation by vitamin D. A molecular machine assembles at the gene. This is RNA Polymerase. It locks on and begins to read. It moves along the strand, building as it goes. This orange thread emerging is RNA, the genetic code transcribed from DNA into RNA, the first step in building every protein your body needs. Look closely. Each letter, T, A, G, C, is read in sequence. The machine never skips, never guesses. The code is absolute. And again, this is real time. This is the actual speed of data being written inside you. One RNA strand complete, carrying its message outward to become something real. The double helix, 2 strands bonded together, holding 3 billion letters of genetic information. When the cell needs to read a gene, only one strand serves as the template. The other waits. 4 chemical letters: thymine, adenine, guanine, cytosine. Every instruction your body has ever followed, written in combinations of just these 4. Each rung of this ladder is a base pair, and your genome contains 3 billion of them. A library so vast it would fill books stacked higher than the Eiffel Tower. But the cell cannot leave all 3 billion base pairs open and exposed. It needs to manage access, so it wraps the DNA around proteins called histones. This is a nucleosome, 8 histone proteins with the DNA wound tightly around them, like thread around a spool. When DNA is open and unwound like this, it is active, accessible, ready to be read. The genes here can be transcribed. Those small orange marks you can see, those are epigenetic tags, chemical signals added to the histone proteins. They tell the cell, this section is open, this gene is on. But other tags send the opposite signal. They cause the DNA to coil even tighter, locking it away in dense, unreachable fiber. That gene goes silent. Active and inactive, open and closed. A constant, delicate balance, written not in the code itself, but in the way the code is held.